Hello, everyone, and today we'll be discussing the final project here, completing labs 4 through 7 and connecting all of our pathways here. We'll discuss the formation of ATP through several pathways, as well as some other cofactors and intermediates that can be used within the different pathways of each lab here and within the different cycles. We'll talk about how we will eventually lead up to our ribose through some of our pathways that use intermediates from glycolysis, and we will discuss the pyrimidines and the purine formation through use of some cofactors as well as our PRPP. Okay, so there's many ways that we can take in certain molecules throughout the body or produce certain molecules throughout the body and use those in certain pathways to create ATP. And so we can start off with sugars or fats that we can take in through the body and through the formation of, or through the intake of dietary fats, we can form glycerol, which can be used in a few pathways here, gluconeogenesis or the glycolysis pathway through the formation of glycerol phosphate with the synthesis of dihydroxyacetone phosphate. Okay, and so by using glycerol here, we can form some other intermediates such as, again, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, as well as acyl-CoA and acetyl-CoA, which can ultimately be used in the citric acid cycle down here. Okay, so through the process of the citric acid cycle, through the use of acetyl-CoA, we can use oxaloacetate to form alpha-ketoglutarate, which will produce some NADH molecules and ATP that can eventually be used in our electron transport chain here. And it's also important to note that within the citric acid cycle, some intermediates can also be formed through these amino acids or through the formation of, or through the use of alpha-ketoglutarate or NADH and NAD molecules or cofactors. So here we have alanine, which can be transformed into pyruvate, aspartate into oxaloacetate, or glutamate into alpha-ketoglutarate, all intermediates which can be either used in the citric acid cycle or in the pyruvate dehydrogenase complex, such as pyruvate, where we decarboxylate pyruvate and get it ready to enter the citric acid cycle here. So through the intake of dietary fats and the use of acetyl-CoA and glycerol, as well as some amino acids, we can create some ATP and some cofactors which can then be used to enter the electron transport chain. Okay, now let's consider the use of glycerol 3-phosphate in the formation of triacylglycerols or triglycerides here. So through the glycerol 3-phosphate, we can create our intermediates such as diacylglycerol and 3-hydroxy-3-methyl-glutarol-CoA. And so keep in mind, this is another intermediate that can also be formed by the use of acetyl-CoA up here after using our glycerol that we take in from our diet here, okay? And so another important cofactor to note is our dimethylallyl pyrophosphate, which can also be used down here as an intermediate to form squalene, which will ultimately form cholesterol through cholesterol synthesis. And so as you can see, a lot of these intermediates are tied in through many pathways and can be used, especially cofactors like the ones generated here in the citric acid cycle. So after these these electron carriers are transported over to the electron transport chain, we form the proton gradient, which fuels the mechanism of the ATP synthase enzyme here. And it's through that proton gradient and the electron transfer from the electron transport chain that we can create more ATP, the same way we do in the pathway of glycolysis. So the same way we can generate ATP from the intake of dietary fats and the use of glycerol, it's the same way we can generate ATP through glycolysis by the intake of sugars like fructose, sucrose, and the use of glucose 6-phosphate, which will ultimately be sent and used up here to form our ribose 5-phosphate to obtain our PRPP, which will then yield our pyrimidines and our purines here and our nucleotides. So through the process of glycolysis, we end up forming NADH and using some ATP, and the formation of pyruvate can also be used down here in the pyruvate dehydrogenase complex, as well as the NADH formed here in the electron transport chain. So the same path that the cofactors of the citric acid cycle take to form ATP is the same pathway that these cofactors here take to form ATP through the electron transport chain. So after this ATP is used, we can use this, after this ATP is formed, we can then talk about the glucose 6-phosphate that's eventually used here in our ribose synthesis here. And so there's many sugars here that can be interconverted and used, even intermediates like the GAP molecule that eventually will lead back to over here, our glyceraldehyde 3-phosphate. And so our glucose 6-phosphate here will eventually be synthesized into ribose 5-phosphate by the use of the PRPP synthetase here. And here is where we eventually transfer our PRPP over here to this pathway right here. Okay, and so we can create our nucleotides here, our UTP, CTP, and TTP, or our purines here through the use of our carbon dioxide or the PRPP that's formulated through this pentose pathway over here. Okay. Oh, and it's also important to note that lactose and galactose can also be synthesized to form glucose 1-phosphate, and that will eventually yield our glucose 6-phosphate through the use of our phosphoglucomutase enzyme. Okay. So through the use of our carbon dioxide, the formation of our carbamic acid can lead us to the formation of our uridylate, which can then be used with different monophosphate kinases, which can be transformed to UTP. UTP can then be used to synthesize CTP, and through our UDP, we can then transform through the thymidine synthetase to TDP, forming our TTP molecule here. Now, if we go back to our pentose pathway here, our PRPP can be used here in this pathway. And so by forming our inosine through these intermediates here with our ribose, we can then use the inosine to form our ATP through fumarate, through the formation of fumarate, or by the use and formation of NADH, we can form our GTP here. Okay, and it's also important to note that the formation of methionine or methionine can be used to create our succinyl-CoA here, but it's through many intermediates and many formations here. All right. So we've discussed how we can generate ATP from taking in sugars and fats and using glycerol to send to our citric acid cycle. We've talked about our succinyl-CoA formation through different amino acids, as well as the use of our dimethyl allyl pyrophosphate in the formation of cholesterol to store, as well as the transformation of alanine, aspartate, and glutamate to form intermediates that can also be used in our citric acid cycle. And all of these eventually forming the ATP products used or formed from the electron transport chain, as well as the ATP synthase molecule. All right, thank you very much.